Okay, let's get started and go back to GlyGen. You already saw the front page. Karina was talking about that, and we don't just have glycans in there. We also have proteins and, of course, the glycoproteins in there. We had, before I forget that, we had a question in the chat about the recording. Yes, the recording will be shared. Also, the recording from last week. It just takes us a while to, process the recording, clean it up and then bring it up on YouTube. So just give us some time. We will, once the recording is available, we will send you an email to the email address that you used to register, and we'll notify you once it's available. Okay, coming back to the protein page. Also, we mentioned it in the first session, the way how GlyGen works is it's not like a primary data store where usually people can go log in and create their own account and upload data. The way how we usually work is we acquire data from existing databases about glycans, about proteins, about genes, and we integrate that all in one database, which then allows to browse over all that information in one consistent portal. But we have these links back to the original data sources to give you the possibility to explore these other resources as well. As an exception, as I said before, if you are producing a lot of data, glycoproteomics data, you can contact us and we will work with you to get your data into our data portal. And then from there, it gets integrated in the GlyGen portal as well with the corresponding attribution, like if there's a paper associated with your data or, your name will be associated with your data. Okay, let's go to the proteins. And similar to Karina, I will start with the protein details page. I will also put that link in the chat in case you want to follow on your own screen. And it's a really long page, but I really want to give you an overview. What is all the information that we have in GlyGen about a protein or a glycoprotein? In the first section, in the general section, we have some information like the name, the UniProt identifier, which is more or less the primary identifier for proteins that we use, names or alternative names, mass of the protein, the RefSeq identifier, and also the organism that the protein was found. And then already in that first section, you can see these little buttons here. We call them evidence badges, which link the information back to the original database where we retrieved that information. So for example, the information that protein is encoded in this chromosome in that section came from a sample gene. And if you click on the sample gene, the button, you have a link to that database or that record in that database where we actually pulled that information from, that it's located on that chromosome and the location on that chromosome. And that's true for all the badges that you see on that web page, in the tables everywhere. We are linking back to the original data source that provided that information. And usually these databases have additional information that we didn't integrate in GlyGen because we didn't consider it important. So for example, for UniProt, if you go on the UniProt web page, you will find tons of more information about the protein than you will find on this web page, because we have that, glycan focus, these glycoclasses that we have on and what we want to focus on. But there's obviously a lot of other stuff available, and you can find that on that other web pages. And that's why we have all these links, so you can go there and find that information. Okay, after the general information, the section, or the probably most important section, comes, which is about the glycosylation of that protein. And we have these 4 different tabs here with different groups of information. So the first tab is about reported sites with glycans on it. That means here you can already see the glycan structures, and Karina talked about the cartoon representation that we use, but we also have the glytucan ID and the type of glycan. And then we have the site on where the glycan was reported to be on. And here again, the evidence badges, we talked about that. So there is a database that reported that, that's GlyConnect, but there's also a PubMed, a paper that where you can find that information. And we will get to the paper web page in a little bit. The next tab that we have is, oh, before I move on, you also have these standard controls. So it's 50 different reported glycans on the different sites. You usually just see 20. You can change the account if you want to, but you can also scroll through the page and see more records. Okay, that's a little bit slow, but that's fine. Let's talk about the other tabs. So we have one tabs with reported sites with glycan structures on it. And then we also have that reported sites, which means people just reported that's a glycosylation site, but we don't know what glycans are on there. So if you think about the standard proteomics run, we released all the glycan, we have that scar, that 1 Dalton difference that we can use to identify that's a glycosylation site, but they have no clue about the glycan. Okay, well, I have a bunch of other tabs as well. So if we click on the reported site, it will look like this. Again, the source database, the type of glycosylation, the residue that it was glycosylated on, and sometimes we have additional notes from UniProt that, you know, in this case just says, okay, that's an N glycan. We also have 2 additional tabs which are not filled in these 2 pages that I have open. There is a predicted site, so we sometimes have predicted information either from UniProt or from prediction tools that we run ourselves. And sometimes we have text mining data where we have pipelines to process literature automatically with machine learning tools, and we extract information out of it. And we are not mixing that up with the actually curated information. We have a separate tab for them if for that protein this information is available. What we also have is phosphorylation, because especially like inside the cell, the phosphorylation and the O-glycosylation can occupy the same sites. So also source information, if we know the kinase that added the phosphate to it on this particular position, we will have that as well, and some notes. Glycation information is rare, but we also have that too, where you can find the type of glycation and the residue where it was being reported, and again, the source, the paper, or the UniProt ID. Okay. We went over that. So then we have the name section, alternative names for the protein. These are also names that when you use them in the global search or in the protein search later on, it will also find the protein based on that. The function annotation mostly coming from UniProt and from RefSeq, where you have short sentences describing the function of that proteins. A sequence display, we get back to that in a little bit. The mutation section, and here I should mention it's a single nucleotide variant, meaning it's a single amino acid which was changed. So we don't have multiple amino acids. And it's grouped into either disease-associated mutation, where we know that that mutation on that side of the protein, the K to an N switch, is involved or reported in a certain type of disease. We also, in some cases, have the frequency information. And we have non-disease associated mutation where somebody reported a mutation, but it may not have any effect in the disease or may not be involved in a disease. Mutagenesis information, meaning that's not a natural occurring mutation, but somebody in the lab modified a certain position and changed it. And then usually we have a note there which, you know, explains the consequence, what happened with that change. Followed by that, we have the Go annotation in the free section where we have the biological processes that protein is involved, the molecular function, and the location of the protein where it has been found. If the protein happened to bind a glycan, so up there we had the glycosylation section, meaning the glycan was attached to the protein, but some proteins also bind glycans, and if that is known, we will have it in this section. And again, mostly coming from MatrixDB, which glycan is bound by that protein and in which paper that was reported. PTM annotation are just general comments that curators that, extracted that information from a paper have added to that record. Proteoform annotations, these are certain IDs that have been given to a form of that protein with certain glycans attached. And you can also find the paper information that are associated with that proteoform here in the comment in the description section. But we also have a link to the pro-ontology, which describes that particular form. If the protein was part of a pathway or involved in a pathway, you can find that here. We mostly have sources from the CAC pathway, but also from Reactome. And sometimes you have that show more link, which essentially just hides a lot of the information to make sure that in the beginning, the page isn't all that long. Synthesized glycan, if your protein happened to be an enzyme, it may have been involved in synthesizing certain glycans. And in that case, we will have a list of glycans here where that protein may have been involved. I will show you that for another protein later on. We have the list of reported isoforms, where you can see just a summary of that isoforms, but we also have a little button up here for the show sequences, where you can also show the faster sequences of the isoform that has been reported. Going a little bit down, we have homologs. So that was a human protein, and we have in mouse and rat homologs of that proteins and the source where we got that homolog information. I will come back to that section a little bit later to show you the alignment feature between the homologs. If the protein was involved in a disease or has been reported in a disease, then you will find that in this section. In this case, we only have a single disease associated with that protein. But if there are multiple diseases, you will see multiple of these records. Okay, there was a question in the chat about the SMV section, if we are showing all the SMVs or just the, just the ones, the mutations associated with glycosylation. So we are showing all the mutations that we find, independent if it's related to a glycosylation or not. So unfortunately, let me see here, something like that, right? That has nothing to do with glycosylation. But sometimes, yes, it might actually affect a glycosylation site. And actually, we will have an exercise later for exactly this case. But we are trying to capture all the sites that we can find, as long as they are available in our databases. And let me get back to that, because that's an important point. We are recruiting our information from these other databases. If any mutation that somebody reported in a paper didn't make it into these other databases, we won't have it. Meaning this here is not the complete list of all single nucleotide variants that everybody in the world ever reported for that protein. This is only the sites that we can retrieve from these other sources. And there's a huge gap, especially on the glycan side, where a lot of the information that people have reported, like glycans as attached to a protein, that never made it into any database. And we won't have that information. So what you see here is just what we can retrieve. It's never complete. That's also the reason why we are working with people and want to get their data into our system to close that gap and make it more complete. I hope that answered your question. So let me go back to the disease section. Okay. And then we have the biomarker section. Karina was already talking about that. So if the protein or the glycan has been reported to be part of a biomarker, you can click on that link here and you will go to the biomarker page. Karina already went over that, what you see there. And that's also a very important aspect. All these links that we have here for the identifier, the biomarker, the PubMed ID, the glytukin ID, or the protein ID, we are all leading to other pages in our system. So you can very smoothly transition from the protein to the glycan part and back to other proteins. Let me give you a quick example here. Let's go back to the glycosylation section. So we are on a protein page. These are the glycans that have been reported on that protein, and we have the glycogen ID here. So when I'm clicking on that glycogen ID, we are going to the GlyGen page that Karina showed you. And on that GlyGen page, we also have other proteins which are associated with that glycan, where the glycan was attached. So here's the one that we just came from, but there are also a bunch of other, well, that's the same protein ID. There are other proteins that I can go and look at the information for that protein where that glycan was attached. So it's very easy to, transition between the protein to the glycan, to the biomarkers, to the papers, the motifs, and so on. Let me go back to my page. Okay, we're almost done. So for the protein information, we also have expression information, like the expression in different tissue, where, the expression level, essentially high, medium, or low, was reported for different types of tissue. We also have the expression information in disease, where the protein not just has been reported, where somebody says, oh, that protein is related to a disease, but where we actually have data in BioExpress, for example, that shows that that protein in that disease was, for example, upregulated and downregulated. And then you have also an indicator, was that significant or not? The cross-references, so like I said, for each of the data points that we have always the direct reference, which provided that data point, but we also have that general section where we have all kinds of cross-references. We can expand them all to the pathways, to gene cards, BG, also especially for proteins of interest to the 3D files. And you can see that protein that I picked, there's tons of 3D structures in PDB for that protein already available. And that's really just, this section here is really just to help you find all this information. And like I said, all these databases have additional information, like on PDB, you will find the 3D structure of the protein. In the gene cards, you have more gene-related information than you find on our webpage. Okay. Karina mentioned the history section. That's just to keep track of changes in the record. So this one was added in 1.113 to GlyGen, but sometimes a protein record gets merged or they get split up into individual records. You will also find that here. And lastly, you have the list of all papers that we have for that particular protein. So that's just to give you an overview, tons of information on that web page. And obviously, you don't need to scroll like I did. You can also use that left side menu to jump to the particular section that you are interested in. So before we move on to the actual search feature, I want to point this feedback button on the right side here to you. So we are very interested in getting any feedback. And as you have seen, sometimes something doesn't work, or sometimes something isn't correct. If you see something like this, don't just say, oh, this is rubbish and go away. Just tell us about it, okay? You can use that feedback feature here. You can report a problem. You can make a suggestion. You can ask a question here. Just put down your name and your email address, and that will go into our system, create a ticket for it, and then we will respond to that, either by fixing a problem or, either following up on a suggestion or explaining why we don't want to do that. So this is really a system that we developed for you guys. So tell us how we can improve it. Okay, so let's talk about the protein search. I'm going back to the homepage. So when you open that glygen.org link, you will be ending up on that web page, and you have two options. If you want to start with a protein search, actually free, you can use that global search. We shortly talked about it before, and I will show you another example later on. Or you can click on that protein search card right here, or you use the menu, Explore, and Protein Search. And similar to the GlyGen search, you have that simple search where you can just type any text you want, the name of a disease, gene name, protein ID, a pathway, whatever, and then perform a search, and it will show you what it finds to it. And we also have that advanced search feature where you can really pinpoint more exactly what you're searching for. So for example, you can search for particular accessions, or for example, if you did a proteomics run, you may have a bunch of protein IDs that you copied from a table. You can also drop them here, all of them, and it will make a search for all these proteins, protein accessions. You can also use RefSeq accession. We talked about a mass search, probably not so useful for proteins. For glycans, it's more useful. You can restrict your search for certain species. For example, all the human proteins, protein names, gene names, Go annotations, Go IDs. Can also, if you have a certain glycan and you want to know if that glycan is bound by a protein. And we are not talking about glycosylation here. We're really talking about binding. So if you have a glycogen ID, you can say, okay, find me all the proteins that are reported to be binding this glycan. Then you can obviously search for glycosylation type, either just, plain NO glycosylation type. You can also say which glycan are you interested in? Find me all the proteins where this glycan has been reported on. You can also restrict it by the type of amino acid that has a glycosylation information on it. And you can actually select multiple of them if you want to. The other alternative is, especially if you're interested in glycosylation, just show me the predicted sites or only the reported sites, or both, which is the default. You can also look by protein sequence. You can either drop an entire protein sequence, you can also drop a peptide sequence, or you can drop like wildcards, like something like this, NXT, which is, the consensus sequence for N-glycosylation site, and it will find all the proteins which contains that short peptide, in this case with that placeholder. Pathways, disease name, disease ID. You can also look if you have a paper and you want to know if we have the information of that paper in the database. You can also put your PubMed ID in here, and it will show you, first of all, do we have that paper? And if we have that paper, which ligands or proteins have been reported in that paper? For biomarker, you can also search for all proteins which have a biomarker association for a certain disease, or, based on the biomarker type, like is it find me all the proteins which are diagnostic biomarkers or monitoring biomarkers. So let's just do that search. So the only two features that I enabled right now was I selected human and I put that NXT down here. And then if we perform the search, it takes a second or 3. And then we get the results. So we have 11,000 protein sequences here. We can see in that box up here what was our search criteria. Karina showed you before, if you click on modify, you will get back to the search page with that stuff pre-filled, and you can modify your search query. Another way how you can shrink down the list of proteins that you have on that page is by using the filter option. We searched only for human proteins, so obviously human will be the only feature available, but we can now also filter by glycosylation site. So for example, only show me the N-glycan, or show me proteins with reported N-glycosylation. So we are now down to 2,000 protein from the 13,000. You can also filter proteins by the biomarker type, the size, which probably is not very useful, but you can also filter them by the data type. So for example, do we have, show me only the proteins which have disease information associated with them. So if you click on that, our 2,000 proteins that we just had goes down to 750 proteins. And we can keep adding more criteria till you are happy with the list of results that you have. So a few more things on that page, and then we go to the first exercise. Karina mentioned that the hit score. So that kind of score that you see here is not like a probabilistic score or something like that, which says, oh, that's most likely the protein you're looking for. So the way how this works is it searches for the protein or the glycan based on your search query and your filter options. And then essentially it rates them with for the information that are associated with that protein. So you can see that in the matrix, we have different pieces of information. Each of them has a weight and has a frequency. So for example, we have 400, for this particular protein, we have 400 GlyGen information reported for that protein. There's a certain weight to it, and that adds up to a score. And the higher the score is, the higher it shows up in that list. So that essentially means that the protein with the most information in the system will show up in the list first. So just as a warning. One more thing I should mention, you can sort all these columns. By default, it's sorted by the score, but you can also sort it by the ID, the organism, the RefSeq ID, or the protein name. The other thing I would like to mention is the download feature. We touched on that before on the GlyGen section, where we said, okay, you can download this entire list of GlyGens into a Bionic database. For the proteins, you can download this table in a CSV file. And if I'm clicking here, it will, calculate for a few seconds, and then you can you get that file that you downloaded onto your computer, and then you can work with that. And that file will have pretty much the same information that we have in that table here in the beginning, and then some additional information towards the end. And that's the entire table. What you also can do is you can download all the FASTA sequences for all the proteins in that file into a single file, and you get a machine-readable JSON format if you want to. Okay, there is a question if you can control the columns in that download file. Unfortunately, not right now. So, but you saw that example file. Honestly, once you have it in Excel, it's not a problem to delete a column. One of the things that we are planning in year 3, which is, the coming year, is to allow users to customize that table and, remove columns and add columns. And then that will also affect the download feature. But that's on our list for the next year, essentially. Okay. And we talked about the modify button already. So now what I would ask you to do is go to that folder with the handouts and open the PDF with the protein and try to solve exercise number 1. I will give you 3-4 minutes, and if you're interested, try it out yourself. I will show you afterwards how you can do that. So the exercise is the following. Find all human proteins that have both reported O-glycosylation sites and phosphorylation sites and have reported mutations. So like I said, I give you 2-3 minutes to try it out yourself, and then I show you the solution for that quiz. And if you have questions, feel free to ask them. Okay, no questions. I'm not sure how many of you tried that little exercise. But you can also download the PDF later on and take a look. And it really has a step-by-step instruction with screenshots how you can solve that little question. There is not one way. Actually, for that particular one, there are multiple different ways how you can approach that. I will just show you how I would do it. So the question was, first question was all human proteins. So that's an easy pick. So you just filter, we make a search for all human proteins. And since I know the system already, I know that I can solve all the other problems with the filter options on the search page. So I will just search for the human proteins, and that's it. And we will end up with, more than 20,000 proteins in that search option. So there was the question for the SNV sites. So in the data section, we can select all the proteins which have SNV mutation reported for them. And that will bring us down to 11,000 different proteins. And then the other section was we wanted to have proteins which have both O-glycosylation and phosphorylation sites. So let's go for the O-glycosylation first. That will shrink the number down to 6,000. And then we click on the phosphorylation site. And now what happens here is it went up to 8,000. Now, the reason we would actually expect it to be lower, right? But the reason why that happens is because all the operations in that field here are all connected. So we didn't ask the question, find us the protein which has both O-glycan and phosphorylation site. We asked the question, find all proteins which have O-glycosylation or phosphorylation sites. But we can change that just by changing that drop-down. And now it will really combine all the information in that box with an AND, and that brings us down from the original 6,000, which had O-glycosylation site, to 4,000, which have now both O-glycosylation site and phosphorylation site. So that filter option is really very powerful. Between the different boxes, it's always an AND connection where we say, okay, whatever you selected here and whatever you selected here. But within the fields, you can also choose if you want to have an OR, like we said before, which have either O-glycosylation site or phosphorylation site, or you can make it AND where you say, okay, it must have O-glycosylation and phosphorylation. So that's really a very powerful mechanism to shrink down your search results to to what you're actually looking for. Now, the last way, and we had shown that before, how you can search for anything, glycans and protein, is you can use that bar up here, that search field. And in my case, I would just look for enzyme, the SD3-GA6. And it will perform that search and will find anything, any record in our system which is somehow related to that SD3-GA6. So there are around 2,000 glycans, which were that it's an enzyme, so it's reported to have modified these glycans, 2,000 structures. And we have we have 17 proteins which are related to that enzyme name or to that name somehow. And if I'm clicking on that, you can see, okay, the first three annotations is essentially that enzyme, the one in human, in mouse, and in rat. And then there are other enzymes which are related to that because we pretty much have the same function, or a similar function, not the same function. And now let me just quickly click on that protein here and show you one of the things that I mentioned before. So for proteins that are actually enzymes, glycoenzymes, you can go to that synthesized glycans section, and you will find the list of glycans where that protein might have been involved in making that glycan, which means it's more or less in this case about adding the sialic acid to the structure. And you can see these are the 2,000 glycans that we talked about before. Nathan will talk about that a little bit more in his talk about the sandbox next week, like how this annotation was added to the glycan structures. So if you're interested in this and also about the glycosylation pathways, you may want to join Nathan's talk next week. Okay, so let's close that and see. Okay, now back to a protein page, and in case you want to follow, I will drop that link for that protein that I have chosen here into the chat, and you can follow along if you want to. On the protein details page also, we have a download option here. The default is you get that machine readable data file, which is more or less of interest for you if you're a bioinformatics person. But we also have automatic APIs, so you don't have to, if you have an MS run with 500 proteins, you don't have to open 500 browser tabs and download 500 files. There's also APIs where you can automatically retrieve that information if you want to extract information from our system. And Robert will be talking about that in 2 weeks. Another way how you can download the protein is in a FASTA file, which is the FASTA sequence. And then you can use that for whatever tool you want to use it for. We talked about the sequence option. Yeah. Okay. Now let's talk about a few tools which are integrated in a web page. And we are back to the glycosylation side, glycosylation section of that particular protein. And any section which has like a table-like format, you always have a download link where you can download this table into a table on your local system. I should warn you, for the glycosylation side and also the glycation, you will not get the images in your Excel table. You will just have the text columns, but you can download them into that format. And for that particular section, since we have like different tabs here, the reported sites with glycans and predicted sites, you can download them individually. Okay, so let's talk about tools. So one of the tools that we have integrated in the web page is called ProtVista, which is actually developed for UniProt, but we took that tool and we modified it for our purposes. Let me just change the screen size a little bit. So on the top, you can see the navigation pane. So it's amino acids is from 1 to 1207. And you can also use that to navigate down to a certain section of the protein that you are interested in. Below that, we have different bands where we have information in there. The sequence band, that's usually too crowded, but if you're scrolling down to a certain section, the protein sequence will start to appear. Then we have the glycosylation section where you have different symbols here. And when you mouse over some of the symbols, like this one, you can see in the headline the site. There's a serine at 317, and you can see 377, and you can see the glycan structures or one glycan structure that has been reported on that site. When we have multiple glycan structures that have been reported on that site, you will also just see one representative, but then it will have a text below it saying 10 more glycans reported on that site. So you can also expand these bands, and you can see the different information split up in different bands. So here we have end glycosylation site. There was a composition reported on that site. We have much more glycan sites, like just sites information where no glycan has been reported for that site, just information. It's an end glycosylation site. We have the same for O-glycan, where we, in this case, we only have sites with O-glycans attached. And then we have the places for the consensus sequence. Now, you cannot just mouse over any of these symbols. You can also click on them. And if you do that, you end up on the site view page. So this is the page for this protein on that particular site. Okay. And Jeet Vora will talk about that next week a little bit more. But this page will give you detailed information about everything which happens on that particular site, which, that's the glycan that we just looked at. Like I said, Jeet will go over this in detail next week. And you can also ask him questions there. Also, the other information like phosphorylation, so there is one site reported for that protein, or glycation information, nothing for this, or the mutation information, you can see everything represented by individual dots here. Okay, let me go back to the protein side. So that's one of the tools, and you can see pretty much any section that has anything to do with sites, like glycosylation, phosphorylation, glycation. We have that ProtVista tool where you can go there and see that overview. Now, another way how you can explore that is the sequence viewer that we have on our side. So this is the protein sequence. And then on the right side here, you see all the different annotations that are there. So for example, that protein that we have here has 9 end glycosylation sites. And if I tick that box, it will highlight the end glycan sites. And we also can see that we have 9 sequons here. And well, no surprise, it overlaps with the N-glycosylation sites. But we can also add other annotations like phosphorylation or mutation here. And then we can see where it overlaps with that annotation. So one of the questions before was, do we have mutation that is outside of glycosylation sites? And if I just add the O-glycosylation sites too, you can see, okay, there's a lot of mutation which has nothing to do with the glycosylation sites, but sometimes, like in this case, it might affect the glycosylation site where either the asparagine gets mutated, and that's gonna modify the glycosylation site, or, you know, from the consensus sequence, that residue gets modified, and that might also affect the glycosylation. What you also can do is you can highlight certain amino acid sequences. So we talked about that the NXT is one of the sequence, or we can also say the NXS and the other sequence, and I will highlight this short peptides that you can enter here. So at that point, I would like to do the second exercise. Again, the Google folder is still in the chat, and I will drop it again. And that exercise is essentially about that sequence viewer. And what we want to do, oh, sorry, it's for other protein. Let me just open that link for that protein and also put that into the chat. So it's about that protein that I just put into the chat, but you can also, you know, open it when you click on the link in the PDF. And what we want to do is a continuation from the search that we did before. In that particular protein, find all sites which are co-occupied by O-glycans and phosphorylation. So sites that are both O-glycosylation sites and phosphorylation site, and that may have been disrupted by a mutation. So again, I will give you 2 or 3 minutes to, you know, open that page and play around with it yourself, and then I will show it to you on my screen. Okay, no questions so far. So let me show you how one way how I would solve that problem. Again, there are multiple ways how you can do that. I mean, the most boring way would be you could go to the phosphorylation section, go to, on the table, you know, write down all the sites, and then you go to the glycosylation section and you write down all that sites, and then you go to the mutation and you write down this sites, and then you compare it. Or we can use the sequence view, if we talked about before. So we wanted to have all the sites which are O-glycosylated. So we select O-glycosylation sites and phosphorylated, and you can see, okay, there's a lot of phosphorylation going on here in the beginning, and you can see it also we co-occupy certain residues. Of course, you know, the phosphate and the glycan cannot be at the same residue at the same time, but you know, sometimes it's phosphorylated and then sometimes it's glycosylated. And sometimes it actually gets switched, you know, during the life cycle of the protein, where the phosphate or the glycan actually work as a switch to activate that protein or deactivate a certain function. Okay, and now we want to see, okay, which of these sites are affected by a mutation, and we have the mutation right here, and we click on it, and it looks like, oh, there's this place, but nothing else. Well, the problem here is both of these highlights use essentially highlighting the background of the character, and the phosphorylation paints over the mutation. So if we just toggle the phosphorylation on and off, we will see, okay, there's a lot of more mutation going on, and also on the side where the phosphorylation is on there. So that's just something to keep in mind if you're working with, you know, the highlighting where it highlights the background of the residue. It may actually overpaint with one of the other options here. Okay, but what we can see is, okay, there are multiple sites on that protein which have mutation information, which, you know, may disrupt the glycosylation and phosphorylation of that protein and then have certain effects on the function of that protein. Okay, a few more things that I want to show you. Let me go back to my other example protein. So we already talked about, we have isoforms, and we have the isoform sequences, and we have the homologs, and we also have obviously the homolog sequences. But one of the things that you can do is you can align the homologs and also the isoforms. I will just pick this database where we got the homologs from. Then I click the alignment button, and it brings us to this kind of alignment view. Most of you are probably familiar with that. But what you can also do in this view is you can use the same option that we saw in the sequence viewer, and you can highlight the glycosylation sites. You can see them here, the N-glycosylation site, the O-glycosylation site, and all the other things like the mutations. Mutations in this context is not so much of important, but we can, for example, see that this glycosylation site, or this one up here, down here, and down here, they are actually reported in all the homologs. So that might be an important site because, you know, it's there in all the 3 different species, while the O-glycosylation site is only reported for human. Now, here comes back what I said earlier. It doesn't mean that in mouse or rat, this isn't actually glycosylated. It may just not have been reported to be glycosylated yet, or it may not have been put into the database yet. But that might give you an idea about something, you know, if you are working on mouse and rat, something to look into. You can do the same with the isoforms of the protein, where you can align them with each other and then highlight the different isoforms. The last thing I want to show you, or one of the last things, I have many last things I want to show you, is the publication page. You saw there's a publication section in the end of the page with, you know, all the papers that are talking about that protein or that glycan. You have the same section there, but you also have PubMed annotations for certain other information like glycosylation. When you click on the PubMed ID, you're not actually ending up on PubMed directly. You're ending up on our publication page for PubMed. And then in the top section here, okay, we have the title, we have the authors, we have the journal, and then we also have the link to the actually PubMed ID, and then you go to PubMed for this one. But then what this page also has is a summary of, you know, what was reported in that paper. Like we were talking about human stuff in there. We were talking about these proteins. We were talking about a bunch of glycans. And we were even talking about glycosylation in that paper where on this protein, this glycan was reported on that site. So you can also get an overview about the content of certain papers. And a lot of the sections are usually empty because you hardly have a paper which talks about all this stuff. We only talk about, you know, per paper, one of the aspects. Oh yeah, and we also have expression information for glycans in these papers, and then you will find out that this glycan on this protein on that side was, for example, expressed or found in a certain tissue. And sometimes you also have abundance information, although that's really rare. Okay, talked about that. I wanted to show you one more thing. Karina was talking about the motifs where, you know, you go to a glycan page and then you can use the feature viewer to highlight certain motifs, or you get the list of motifs. And then by clicking on the motif ID, you go to the motif page. If you're interested about, you know, all the motifs, what kind of motifs do we have in GlyGen, you can go to the Explore menu and then go here to the list of motifs. And that gives you pretty much the entire list of all the motifs that we have in our system. You can see the motif ID, the common name for that motif. If there are certain synonyms for that motif, you can also find them. And you can find the number of glycans and proteins that motif has been reported. Obviously, the protein doesn't contain the motif, but the protein has glycans attached to them, which have the motifs. And also you can download this table into the entire motif list into a JSON file.